origin of life in Precambrian evolution. Then we're going to talk about the macrobiotic life after the Cambrian explosion tomorrow, and we're going to talk about human evolution sometime next week. These are all things that are kind of interesting to know and think about, but they're not actually super specific processes and concepts, sort of unlike the rest of the class. There's these ideas and concepts about how things ought to work and that kind of stuff. This is kind of a tour of how things happened to have gone so far as scientists can reconstruct. So it's going to be a little more historical, a little less conceptual, probably. I try to add as much conceptual stuff as I can. It's kind of weird to have humans be the last one. It's kind of as though evolution like builds up to talking about humans. That's kind of stupid because we're kind of irrelevant to the process. But anyway, that's what we're doing. So without further ado, here's the history of everything as currently understood by science. And the great thing about science is you revise your understanding of how you think things worked based on additional data, additional evidence. So those of you who are astute and well-informed will see that this version of the book says, whoops, I've got to push this button first. The universe begins with Big Bang 16 billion years ago. The estimates now are 13.8 billion years. So just within the time course of a few editions of your textbook, these kinds of things, and there'll be lots of numbers and dates that we kind of throw around, they're really not the essential things for you to learn. There are some of these dates and some of these major transitions in the history of life that you'll want to know what they are and have a general sense of the time scale of when they occurred. But these things are scientific estimates, and they change. So here we go right off the bat. The universe begins with a Big Bang. Approximately 13.8 billion years ago is a current estimate. Let's see. The solar system condenses. The Earth is made. It's bombarded by all this stuff. A giant thing smashed into the Earth and broke off this huge chunk. That's where our moon comes from, being smashed off of the Earth. The oldest terrestrial rocks that are identifiable, found now, known ones, are about 3.8 billion years old. And there's really nice, strong evidence of life by roughly 3.5 billion years. You get this oxygen atmosphere by about 2.5 billion years. So we're talking about an enormous time span of Earth history that we're dealing with. And this kind of strange-looking phylogeny in the middle is intended to show you something about the complexity of what we think happened in these early life evolution in all these different lineages. And there's a couple of things that you'll see on this phylogenetic tree that you're not really seeing on lots of the other ones. One is, let's see if I can, no, stop, go back. That's what I'm trying to do. One is that you have these branches that go off of one lineage and then they connect to another one. You haven't seen that in any of the phylogenetic trees that you've seen before. You always have seen these trees that split into ever greater number of branches. You don't have things that apparently connect one lineage over and into another one. And we'll talk about that as a feature of organismal evolution later today. Then the other thing you see when you look at this kind of scale history of life, you see a whole ton of these branches stop. So you got a whole bunch like this layer here. So a branch that stops is just an extinction event, right? Some lineage went extinct and is no longer there. But you have these periods of time where a whole bunch of branches stopped all at once basically congruently. This particular one is what's at the end of the Permian era. And there's other ones that we'll talk about 65 million years ago when the dinosaurs went extinct, where you had a huge, almost a complete turnover of the large-scale fauna on Earth. Most of the, well, I don't know about most, huge turnover. Let's just leave it at that. Okay, that's the slide I added by mistake. So the kind of time frame we're talking for the origin of life is not known precisely. So we have this age of the Earth, roughly 4.5 billion years old. We're reasonably confident that life existed by about 3.85 billion years ago. And the reason, this is sort of an at least kind of estimate, because the oldest known sedimentary rocks contain traces of life. There aren't older known sedimentary rocks that are known to not contain traces of life. So perhaps if older ones are found, they will also contain traces of life. Who knows? Maybe they won't. The evidence for these old rocks containing traces of life is really based on the ratios of stable carbon isotopes. So living organisms preferentially use carbon-12. So as organisms take in carbon from the environment, they're taking in the ratio of carbon that's typical to the atmosphere, carbon-12 and carbon-13, but they use carbon-12 preferentially. So living or previously living organic matter has more carbon-12 relative to carbon-13 than you would expect from random acquisition of carbon from the environment. So that's some of the evidence. So here's what they are. They're from these rocks on Greenland. The age of the rock is dated by radioisotope dating, and inside the rock there are these apatite crystals, and there are carbon specks inside of those that have this carbon-12 to 13 ratio typical of living organisms. So in part that it's this completely embedded within the rock, embedded within the crystals, makes it unlikely that these carbon sources that have ratios typical of living organisms is some kind of contaminant. Right? You could always find a really, really old rock that has some kind of more recent living stuff in it because it's been contaminated and added later. So the fact that these are completely embedded within the rock structure is one of the things that suggests that that those carbon specks are as old as the rocks are. So how did this start? Obviously no one knows. But there's kind of two alternatives that seem possible. One is that these organic molecules that are the building blocks of life came from somewhere other than Earth. So if it's not Earth, that's called extraterrestrial. Extraterrestrial doesn't just mean little green spaceman, right? The Earth is the Terra, and if it's outside of the Earth, it's extraterrestrial. So that suggests that these building blocks were created chemically somewhere else and got here, and the alternative, of course, is that they started here. Those are kind of the two conceivable alternatives. And we're not really going to try to distinguish which of those is more likely. Obviously, organic molecules could have both arisen on Earth and have arrived on, dust of comets and asteroids and stuff like that. Either way, as a sort of process of thinking about science, you would have to be able to conceive of mechanisms of synthesis of organic molecules from inorganic molecules somewhere, either on Earth or not on Earth. Now, it has to be plausible. So getting these kind of organic molecule basic building blocks from inorganic compounds was studied a lot. It's still an active area of research. Probably the most famous experiments with this was done by a guy who was a grad student at the time, Stanley Miller. he boiled these inorganic chemicals and has hydrogen and ammonia and this electrical spark, looks real mad scientist kind of thing, and completely inorganically, the kind of things you do in chemistry labs, he got amino acids, glycine, alpha-alanine, beta-alanine. In other words, it is chemically possible to synthesize what we consider to be organic molecules from non-living inorganic sources. It's kind of a huge take-home message. And that these can be amino acids and things. And since that time, these kinds of experiments have been replicated in lots of different varieties. You've got lots of amino acids, nucleotides, sugars. It is possible to, under the right conditions, it's possible to synthesize organic molecules from inorganic sources. It's kind of the big take-home message. How people still argue back and forth about the likelihood of that happening on Earth versus somewhere else, in part based on the details of the setup of that kind of experiment. So this kind of thing, how easy it is, depends on whether it's a reducing atmosphere or an oxidizing atmosphere, and these kinds of differences in the different experiments affect the likely outcome or the numbers of different products that are made. So that's some of the ongoing work. What if they came from somewhere else, or both? As I said, they're not mutually exclusive. So perhaps these organic molecules arrived on Earth from somewhere out in the solar system or even deeper space. Some evidence that this is possible. There's this meteorite that hit Earth in 1969 in Australia, and near this town of Murchison. And it was collected and analyzed, and within the meteorite, so again, not like surface contamination, but within the structure of the meteorite, are these various amino acids that are present without any kind of known plausible living precursor that produced them. Who knows what's happening on asteroids? But so both things are certainly possible, that there's terrestrial synthesis of organic compounds from inorganic precursors, and that you get organic molecules arriving on Earth from elsewhere. Does this, to any sort of satisfactory degree, discriminate between these different possibilities, resolve like what actually happened in the case of life on Earth? No, of course not. The importance of this evidence really is that it shows the plausibility of this process, especially if it's replicated over in nearly 1 billion years, 3/4 of a billion years. So these things could have made it to Earth intact, not like cats and dogs flying out of Earth, but the components of organic molecules could have made it. So the more broadly supported probably models that life arose on Earth. This is often called the Operand-Haldane model, and that it kind of outlines a series of steps that would be that would lead to life from non-living sources, and ask kind of what's the plausibility and what's the evidence for any of those sorts of steps being possible, without necessarily accumulating to this is the correct answer or not. But if you have the inorganic synthesis of simple molecules with stuff like the Stanley Miller's experiment, and that they can assemble to larger, more complex molecules, and if those molecules can transmit kind of information, store information about structure, etc., have something that we would say is analogous to a genotype. And if they can do something, as these, you can imagine these molecules swimming around in the ocean or soup or undersea sea vents, sort of scavenging for other molecules, that molecules may be more successful or less successful at acquiring the resources of these inorganic precursors. So if they can do these things, if they have a genotype and a phenotype, then you can get variation and selection and kind of progressive change. And if you are, if molecules are scavenging other molecules, one way that facilitates that is to increase the concentration of particular chemicals around you. So adding a membrane allows you to do all kinds of things that you can't do otherwise. You can concentrate chemicals in excess of what they're found in the environment. You can have osmotic gradients. You can have all kinds of things that would allow for energy sources and so on and so forth. So that at some point, if you have a chunk of stuff that takes in energy, utilizes in some sense metabolizes that energy, and creates copies of itself, that's pretty good definition of a living organism. It's something that replicates itself and uses metabolic energy to do that. Right? Try to think of how would you define life anyway. It's kind of an interesting question. Okay, so here's kind of this schematic diagram of the kind of process where you have these inorganic molecules that get assembled into these kind of simple nucleotide-like organic molecules, and they can store information in catalyzed reactions. They can compete and replicate themselves. And then if you have these membranes, you have some kind of structure that's very similar to some kind of protocell that is competing with other protocells for organic molecules. Not competing because it wants to, it's just that the ones that did a good job of it made lots of offspring, and the ones that did a lousy job at it made few offspring, right? Like everything else in evolution. So, what's the kind of plausibility of assembling some of these simple inorganically produced organic molecules into longer, more complex molecules? How do you get polymers from inorganic synthesis? So one problem is that water both facilitates and inhibits this kind of process. Large polymers, long chains, might have been facilitated by, clays are, so you have big rocks, and then you have small rocks, gravel, and then you have sand. Clays are super, super fine particles, and they're often charged and polarized, and when they line up and dry out and get wet, that polarity of the fine structure of clay can create sort of a lattice work of organizing other kinds of molecules. So this guy Ferris used a clay montmorillonite and used that as a catalyst to join these more monomeric molecules into longer chains of polymers. And when you add additional nucleotides and let it dry out in the clay work, and then you add some more, they're added daily, he was able to produce nucleotide chains that were in excess of 40 nucleotides. So is that what happened? I don't know. No one knows. But it is a possible, plausible process of building these larger polymers from simple molecules. Let's see. So these are kind of like some of the famous starting experiments. Obviously, this area of research, like the rest, is ongoing. People have used different minerals and different kinds of substrates and different kinds of chemical atmospheres to do this kind of work. Some of the exciting work now is based on the idea that these early organisms inhabited seafloor vents and used sulfur as an energy source rather than light or something else like that. But anyway, using a whole variety of different kinds of conditions, there's longer and longer amino acid chains have been produced through completely inorganic kinds of processes. So what would these have to do if they're going to be the kinds of polymers that would be kind of a stepping stone towards living organisms? Is that you would want them to be able to have a genotype and a phenotype, to be able to do something that and variation that affects their relative rates of replication, that affects their ability to acquire additional organic molecules, to build longer chains, and so on and so forth. So one of the questions people grappled with for a while is, what would come first, DNA or proteins? So DNA is fairly inert. It doesn't do anything. It's this great way of storing information in this the sequence of nucleotide bases, but it doesn't have, it sort of has a genotype but not a phenotype. And proteins have all kinds of phenotypic effects. They catalyze reactions, et cetera, but they don't encode transmittable kind of genotypic information. So one of the ideas that people working on this problem came up with is, well, what about RNA? RNA obviously can encode sort of genotypic information. It's a series of nucleotides. Could it catalyze reactions? Can it also have a phenotype? Independently discovered are these things called ribozymes. That is, the word is a contraction for RNA enzymes. So there's RNA molecules that can do stuff. They can break bonds, they can reform bonds, various different ones can catalyze certain sorts of reactions. So there's lots of variation in these RNA molecules and what they can do, but they also have a component of heritability because of their sort of genotypic structure. So this kind of work led to what's now called the RNA world hypothesis, which is that in the development of life on Earth, RNA came first. So that as an information molecule, as well as this kind of proto-organism, that RNA was what catalyzed reactions and encoded genotypic information, and that only later was DNA used by organisms to encode it. Those things were separated, so that DNA was used to encode genotypic information, and RNA was used as this intermediate kind of messenger molecule. So the idea here is that you had an early history of living organisms that used RNA as their information-carrying molecules, as their genotype. And they have those lineages, or, well, one of those lineages switched to using DNA, and that organism is the most recent common ancestor of all the stuff that's alive now. All stuff that's alive now uses DNA, except for some viruses. And that these extinct lineages of organisms, that was this early world was RNA based. So why are people leaning towards this kind of hypothesis? Obviously, these ribosomes have a phenotype and a genotype. That's important. They have many more functions than just like breaking hydrogen bonds and reforming kind of bonds. There's now, a whole variety of ribosomes that are known to do a whole variety of different things. Phosphorylation, amino acid transfer, peptide bond formation, carbon-carbon bond formation, substrate specificity, which is important as kind of enzymes, right? So you have this whole suite of different RNA molecules that can do different kinds of things and encode the ability to do that based on their nucleotide sequence. Question is, could RNA molecules evolve in a way that is directly analogous to evolution by natural selection? In other words, could you have variation among these ribozymes, and they're in some sense competing for some substrate or some inorganic molecules? Could they become better and better at doing that? That's evolution by natural selection. Can they evolve? So, I think this is just what I said. Can they show adaptive evolution, basically? Could you have whole lineages of RNA that are competing and showing non-random progressive change due to natural selection? Remember, natural selection is the non-random component. So here's this kind of complicated experiment that used RNA molecules to and generation time to drive the evolution of these RNA molecules. What's weird about this experiment is that the adaptive changes that happen in the RNA molecule are adaptive only in the context of this experiment. So basically, the researchers selected for fast generation times. And fast generation times depends on how quickly you can reproduce all of your molecular machinery. And one way you can reproduce your molecular machinery faster and faster and faster is to have less complicated, shorter polymers. So what they observed over a number of serial transfers, which are analogous to generations, is that the total amount of RNA made is not this linear function. It tapers off. So the RNA molecules that win in this kind of evolutionary contest are ones that are shorter. And that's, if you think back to life history evolution, if you have this fast generation time, high risk of extrinsic mortality that favors early reproduction, short generation times, this kind of thing, that's the kind of adaptive evolution that is happening in this experiment. Now, why I say it's weird is that the natural function of this is that it's this RNA affects the ability of this bacteriophage to attack bacteria. So the true function of this molecule is to facilitate the bacteriophage getting into bacteria, and when they selected for faster and faster generation times and got shorter and shorter and shorter molecules, they actually became much worse at infecting bacteria. So that function was, which would be the normal function, was kind of destroyed by the experiment in preference because of the short generation time selection. So it's adaptive in the context of this experiment, but it's not adaptive evolution in a kind of whole organism sense, if you can call a bacteriophage a whole organism. But anyway, the point is you can have variation among RNA molecules and exert a selection pressure and observe changes in those RNA molecules over a number of generations in response to selection in a way that makes sense to that context of selection. Might not make sense to other kinds of context. But their RNA shortened by like 83%. It was a very dramatic evolutionary change in this molecule due to this selection. So, what about cells? So a cell has this molecular machinery in it, but it has this compartmentalization. That's what kind of defines a cell, and that's the huge advantage of a cell, is this ability to concentrate chemicals and molecules at gradients really different from what is outside in the available world. So there are huge advantages to doing it. Do things evolve just because there are huge advantages if you could do it? No, right? You obviously need a kind of plausible step where each of the intermediates has higher fitness than the precursors, right? You you need a scenario to get there that's plausible and increases fitness, not just based on the end result. So surprisingly, I mean, getting a cell membrane around this molecular machinery seems like a very complicated step, but it's actually biochemically one of the easiest ones. Because if you have molecules that have hydrophilic and hydrophobic ends, they organize themselves into what are called microspheres. They organize into sets of molecules where all the hydrophilic ends are on the outside and all the hydrophobic ends are on the inside. They do that without any other kind of interactions being required. So if you have these sorts of molecules, they spontaneously self-organize into these microspheres, which to a large degree resemble what cell membrane composition. So that's surprisingly not really the hard step. However that works, obviously no one knows. There's very strong evidence that cells existed by roughly 3.5 billion years ago. So this pre-cellular origin of life stuff, the RNA world stuff, there's some nice evidence for pieces of it, and you can kind of logically think through what would be the required steps. And I wouldn't at all try to convince you that scientists have figured out how this happened. But I would at least hope you'd kind of say, well, is that a plausible process? Are there intermediate kinds of steps that have some supporting evidence? What you definitely have, very strong evidence, roughly 3.5 billion years ago, you have these cells that resemble modern cyanobacteria. They're in Chert in Western Australia. You have fossilized things that look like cells. So, what then happens, right? If you have life arising, perhaps entirely on Earth, perhaps extraterrestrial sources, perhaps both, you get to a point 3.5 billion years ago where you have these cell-like things that are the ancestors of everything, right? Eukaryotes and all the other kinds of bacteria. So that would be that most recent common ancestor. And other lineages of RNA and other lineages of other kinds of molecules die out. And you have this sen ancestor, the most recent common ancestor of all living stuff. So, based on you'd like to be able, you can infer stuff about that ancestor from parsimony. But parsimony, you need to understand the kind of phylogenetic relationships among organisms to infer what, character state reconstruction of what those ancestors were like. So this was obviously not much more than a speculative exercise before DNA sequencing made it possible to get the kinds of homologous traits among all living organisms. So if you want to make a phylogeny of all living things, you need molecules that are found in all living things. Otherwise, you're missing stuff. So you need something that's a gene that's homologous in all living organisms. You're talking about going back, across the entire diversity of life. Would you want genes that evolve quickly or slowly? Very, very, very slowly. There's only 4 nucleotides. If you get a gene that changes quickly, you'll have repeated nucleotide changes that would completely swamp out any signal of ancient evolution. So you want ones that evolve very, very slowly. Are those likely to be coding or non-coding? Coding genes evolve much more slowly on average than non-coding ones. Coding ones that do something really, really important are very constrained functionally. You cannot randomly change stuff about really, really important functional genes. You can randomly change stuff about non-coding genes, and there's no phenotypic effect because they're not coding. They don't do anything. So what's used primarily is the small subunit ribosomal RNA, and obvious advantages. All organisms have ribosomes, which is one thing, one of the kind of lines of evidence that suggests this RNA world. RNA was this precursor molecule. All organisms have large and small subunits. In other words, there's conservation of the structural aspects of ribosomes, and they're critically important for what organisms do. What they do is they translate messenger RNA into sequences. So they have this critically conserved function. They're in everything with a critically conserved function, so they should have a really, really slow rate of change. You can sequence them. You can look at a gene tree. So this is sort of a phylogeny. Well, it is a phylogeny, but it's a phylogeny of ribosomal RNA, which to the extent that that matches the phylogeny of organisms would also be the organismal tree. But this is really a phylogeny of a gene. And as you all know, what you see when you do this kind of stuff is three major clusters of organisms, which are the bacteria, the archaea, and the eukaryotes, right? You guys do all this in 106 or 107 or 6th grade. I don't know what it is. But that's not news to you, I'm hoping, right? Okay. So, this five-kingdom scheme that I learned back when I was a wee little pup doesn't capture this the scale of genetic variation among organisms. We use these three domains, things that we think of as really different from each other, like plants and fungus and animals. They, in fact, together possess only a tiny amount of the total genetic variation in ribosomal RNA. So you're actually very close relatives. I mentioned the other day that you share something like 30% of your genes with rice. I mean, all these eukaryotes have very, very similar molecular machinery underlying most of what they do. When did that happen? So, again, no one knows, but there are kind of bookends that you can put around when this would have happened. They had to live before recognizable eukaryotes lived. The ancestor of eukaryotes had to live before eukaryotes. Right? The oldest probable eukaryotes are roughly 2 billion years ago. There's cyanobacteria fossils to at least 2 billion years ago. If those apex chert ones are in fact cyanobacteria rather than just the ancestors, then roughly 3.5 billion years ago. It could be these apex chert cyanobacteria aren't really cyanobacteria, but are this root lineage that then split into bacteria, archaea, and eventually eukarya. So you have kind of minimum estimates. So I guess the answer from what I told you is it happened at least 2 billion years ago, is when, and who knows how much before that. Right? So what would it have been like? Well, the three domains of life now use DNA as the genotypic material. So parsimony would suggest that their common ancestor did too. So that most recent common ancestor had probably made the switch from using RNA to using DNA. It was the end of the RNA world. Let's see. Not only do all three domains currently use DNA, all three domains have DNA-dependent RNA polymerases. So they're without DNA as a molecule, they're non-functional. Now, I mentioned that phylogeny of all living stuff based on small subunit ribosomal RNA is a gene tree, not a tree of species. Whole genome sequencing lets you compare the kind of phylogenies you get based on different genes. So if the evolutionary history of a gene reflects the true evolutionary history of the organisms that transmit that gene into future generations, then you should have concordance among the gene trees for a whole bunch of different genes. But you might have some gene trees that are discordant, right? Where some genes would produce a different phylogeny than other genes. It's going to be the true history of that gene, but just not the true history of the organisms that have that gene. And that's what these lines over here are. The lines that go from one lineage to another are intended to suggest the transfer of genes from one lineage to another lineage. So lateral means sideways. So this is the sideways transfer across lineages of parts of the genetic material. And mechanisms of this can be bacterial conjugation, viruses moving stuff around. There's lots of evidence for this in modern like antibiotic resistance. So you get antibiotic resistant genes developing in one bacterial strain that are passed horizontally to other bacterial strains, not in an ancestor-descendant kind of manner. There's lots of this stuff is known to happen in modern bacterial microbiology. Here's evidence for more ancient lateral gene transfer. So here's a phylogeny, a gene tree of the HMG-CoA reductase gene. So this is the relatedness of the gene sequences for this gene, which is not necessarily the relatedness of the organisms that those genes were extracted from. And what you can see, I hope, is that there's this Archaea globus fulgidus that is known to be an Archaean based on all kinds of other stuff. Hundreds and hundreds of traits and other sorts of genes say this is an Archaea, not a bacteria. But its HMG-CoA reductase gene is much more similar to bacterial HMG-CoA reductase genes than Archaean HMG-CoA reductase genes. So that suggests that the HMG-CoA reductase gene from a bacteria got into an Archaean. So that this Archaean still is an Archaean, but it has within it a bacterial HMG-CoA reductase gene that was laterally transferred rather than inherited in an ancestor-descendant kind of manner. And obviously so we know there's lots of this going around within bacteria, but to have it go across domains from a bacteria to an Archaean is a big lateral transfer across there. What about that same thing, but at a scale not of individual genes? The sort of lateral transfer of chunks of your genome or organisms. And this is related to this idea of with eukaryotes, how did cellular organelles arrive? Again, I'm hoping you've all heard this story. Right? So these organelles are mitochondria, chloroplasts are the major ones. They resemble bacteria in many respects. They have their own cell wall. They have a simple circular loop of DNA. Their DNA is not arranged into chromosomes. They're haploid. The question is, did those organelles arise as internal bacterial symbionts of other cells? Basically, did one big bacteria take in another smaller bacteria that then they live together, and the smaller bacteria inside, that's why it's an endosymbiont, because it's inside, evolved to be chloroplasts or mitochondria, and the rest of the cell evolved to be a eukaryotic cell. Okay? And that's the question. So this hypothesis was proposed by Lynn Margulis long time ago, way before you were all born, based on morphology, membrane structure. And the nice thing about this is, like most of science, is it makes a testable prediction. Right? If this is true, then organelle DNA should be more similar to bacterial DNA than the rest of the eukaryotic genome. And that's what DNA sequencing lets you test, that hypothesis. So here's phylogeny of, these three domains, and what I want you to see is over here is ZMAs, that's corn. I don't know if you know that. That's corn. Corn is a eukaryote, right? It's a big old plant. If you sequence the mitochondrial genes from corn, where do they fall out? The mitochondrial information, that part of the corn genome, is similar to bacteria, not to the rest of the eukaryotic genome. Similarly, chloroplasts come out like cyanobacteria, not like eukaryotes. So that is pretty strong evidence that the ancestor of your mitochondrion was a bacteria, and the ancestor of your chloroplasts, if you happen to be a cabbage, was a cyanobacteria-like organism that photosynthesizes. So you're a mess. You're this combination of different lineages. So a chimera was that Greek or Roman? Who knows their history? I don't know. Greek? Should we take a vote? Let's just say we don't know for sure. It's this mythical creature that is part dog or wolf or lion or something, and part eagle kind of thing. Is this thing that's stuck together, made out of different parts, and that's what eukaryotes are too. You're these organisms that are stuck together out of different parts. Your cells that are near and dear to you are only, partially yours. You have within you these mitochondria that are the descendants of bacteria, and they're chugging away, making kids. At least if you're a female. Males, you don't pass on your mitochondria, so they're stuck. They're at a dead end in you. That's it. That's how the evolution through eukaryotes, single cell eukaryotes.